Okay, can you all hear me? Can you hear me?
Yes.
Okay, I'm going to share my screen and start immediately. Okay, we will continue from where we stopped. A lot of students are sending me, asking me for homework. I told you to finish paper 2 and paper 4 drugs. We finished it in class, but if you didn't, finish it for now until we finish this lesson. Okay, we started this lesson last class. We are still in the core material. And last class, we talked about the anatomy of the plants and specifically the different layers inside the leaf. A quick reminder, the leaf is an organ that is made up of several layers or several tissues. And let me get a picture right there, okay? You have the uppermost layer, which is the upper epidermis here. And this upper epidermis, it does not have chlorophyll, so no photosynthesis happens here. We gave it 3 functions. We said it provides protection and it produces the waxy cuticle that also provides protection, and it is transparent to allow the penetration of light into the deeper layers of the leaf. This is the first layer. The second layer is the middle layer, the thickest layer, called the mesophyll layer. I'm revising, okay, what we took last class. The mesophyll layer, and this mesophyll layer is of 2 parts. You have the rectangular cells that are tightly packed. This is the palisade or the spongy mesophyll. Who remembers?
Palisade.
The palisade mesophyll, and the spongy mesophyll is right below it. The spongy is called spongy because they are irregular and they are spaced with lots of air spaces, and this increases surface area to help in gas exchange. And we said their main function is for photosynthesis. They have many chloroplasts. They are directed just below the sun, right below the sun for maximum light absorption, and the chloroplasts, they don't overlap. And they are very efficient in photosynthesis because they are specialized cells. And we talked about the guard cells, I remember, in the lower epidermis, and we said that they control the opening and the closure of the what? The
Stomata.
stomata. And I remember showing you this moving image. Where is it? Yeah. And we said that the guard cells in the lower epidermis, these are the only cells that have chloroplasts because they do photosynthesis. And when they absorb water while they are photosynthesizing, and I stress on that, photosynthesis happens in the morning because photosynthesis needs what? What is essential for photosynthesis?
Light.
Light. So photosynthesis happens in the morning. This does not mean that in the morning the plants do not respire. They do respire, but photosynthesis is happening more than respiration in the morning, and at night there is no photosynthesis, there's only respiration. Okay, remind me, when the guard cells absorb water, they are, what's the key word?
They become turgid and they open.
They become turgid, very good. They curve outwards because the outer layer is longer and thinner than the inner cell wall, which is shorter and thicker. They curve outwards, and this will make the stomata open. And at night, when they don't photosynthesize, they will become turgid or flaccid. Flaccid, and they don't curve outwards, they fall down, or they curve inwards and they close. Okay, this is what we said last time, and then we ended. We also talked about these points, don't forget them. And then we ended about talking about the minerals that are needed. I just want to revise this part also because they can ask you an essay question about the pathway of the carbon dioxide into the leaf. Carbon dioxide is a gas. So all gases, they move by what? What's the keyword?
Diffusion.
Diffusion. Okay, please give them the whole answer so you don't miss a mark. You will mention that the CO2 will move by diffusion from high concentration to low concentration. The high concentration will be outside the plant leaf. The low concentration will be inside the plant leaf. It will enter through the stomata into the air spaces, and then it will diffuse inside the mesophyll cell inside the chloroplast. They want all these keywords, and the question is out of 3 marks, so we want to score the 3 marks. We said that the nitrate ions are needed to make what? Yes, proteins, amino acids. Magnesium is needed for the making of
Chlorophyll.
Chlorophyll. Phosphorus is needed for cell division and for general health, and the potassium is needed for healthy fruits and healthy flowers. We said all that. We did not say anything new. And we did mention the type of fertilizers. We said the natural, like the sewage or animal waste, and the synthetic or the chemicals fertilizers. They are expensive and they cause pollution, but they are also important. And we stopped here, which includes the remaining or the rest or the second half of the lesson, the experiments. Now, these experiments, I am telling you again, do not memorize any experiments because you can receive those experiments or different experiments. They can come up with any new experiment that you can witness on the exam itself. So the idea is understanding the experiment and from the first minute identifying what is the independent variable and what is the dependent variable. And from there, everything will be fine. Now, in this experiment, what did they do? They got 3 plants. In the first plant here, A, they included all the nutrients in the beaker of water. So here you have all the nutrients. So of course, plant A will be considered or be used as the what?
Control.
Yes, the control. It will be used as the control. I have all the nutrients. No nutrients are missing. So I'm going to use plant A for comparison. In plant B, there was no magnesium. So this plant or plant B was growing in a situation where there is no magnesium. And plant C was growing in a situation or in an environment where there are no nitrates. No nitrates. So what do you think will happen to plant B and plant C? Will they be healthy like plant A?
No.
No, as you can see here, in plant B, there is magnesium deficiency. So what will happen in plant B? Magnesium is needed in the making of what?
Chlorophyll.
Chlorophyll. So will plant B be nice and green?
No.
No, it will not be green in color. The leaves will be yellow. So we will describe it as yellowing. The leaves will be yellow, or there's another name for yellow leaves or leaves that grow without chlorophyll. They are called chlorosis. Chlorosis means no chlorophyll. Yellowing of the leaves. Now, of course, this plant, of course, will it do photosynthesis or it will not be able to do photosynthesis? What do you
Will
think?
not be able.
No photosynthesis and it will die. Now, what about plant C? Will it be nice and healthy or also it will be affected? Come on. Plant A is the healthy plant. Huh? Am I asking difficult questions? I said plant C has no nitrates. Nitrates are needed for what? For the making of what?
Proteins.
Proteins. Will plant C be able to make proteins?
No.
No. So plant C will also be affected and it will not grow like plant A. And this is going to be called stunted growth. Now, I am going to stress on the keywords because you keep on forgetting them in your answers and you lose marks. When there is no magnesium, like in plant B, the plant will be yellow. When the plant is yellow, this means that it does not have chlorophyll, and this is the condition called chlorosis. Please remember the name. Chlorosis means no chlorophyll or yellowing of the plant. This plant will eventually die because it will not be able to do photosynthesis. Is anything hard about plant B?
No.
No. Plant C. Plant C does not have nitrogen. No nitrogen means it cannot do amino acids, means it cannot do proteins, means it will not grow. It will stay small. And this condition is called stunted growth. Those who took year 9, I think they're familiar with the term. Stunted growth means the plant is short. The plant did not grow to the full length. And again, it will die because it needs nitrogen. So plant A contains the magnesium and the nitrogen and all the mineral ions. Plant A, if you notice, it is longer than plant C and it is nice and green. And it is healthy because we gave it or we provided plant A with all the minerals. Plant A is considered the control for comparison. Plant B, no magnesium, so it will be yellow. And this is a condition called chlorosis. And it will die. Plant B will eventually die because it will not be able to do photosynthesis. Plant C does not have nitrogen. It will be short and small. It will not grow. And also it will die. And this is a condition called what? Stunted Growth.
Growth.
Anything hard in what I said? Okay. Now, let's ignore pesticides for now. We'll talk about them later when we study ecology. Fine. Now, this is very important, the leaf starch test, especially the newcomers, because those that took the core understand or will remember, hopefully, the leaf starch test. Now, let's go to the very beginning. How do we test for the presence of starch? If I want to know if starch is
Iodine.
present, I
Iodine.
use iodine. What is the original color of iodine?
Brown orange.
Very good.
Blue
And if
brown.
starch is present, the color should change from what to what?
Blue black. From brown orange to blue black.
Very good. Now, we have a problem with leaves. When I add iodine on my leaf, the iodine will not be able to penetrate the leaf because there is a waxy layer. So I have a waxy layer, an oily layer on the leaf. This will make the iodine unable to penetrate inside the leaf. So I have to do certain steps before I add my iodine on the leaf. Who remembers what are those steps? Let me show you pictures in case you forgot. What is step number one? What is this person or this doing in step number one?
Boiling the leaf.
Boiling the leaf, very good. Before I apply my iodine, if you notice, iodine application is the last step. I cannot add my iodine straight on the leaf because of two problems. Problem number one is that this leaf is covered with a waxy layer, so the iodine will not be able to penetrate on the leaf. It's as if I am putting my iodine on plastic. Problem number two, what is the color of the leaf? It is?
Green.
Yes, it is dark green. And this color, this dark color, will not make me see the color change very well. The color change should change from yellow-brown to blue-black. I cannot see this color change clearly because the leaf is very dark. So I have two problems. I want you to understand with me the problems. Again, I want to do the iodine test. The iodine, the original color is yellow, brown, and if starch is present, it should change to blue, black. I always use my iodine to test for the presence of starch. Now, when it comes to leaves, I have two problems. Problem number one, I have a waxy layer on the leaf that will not make the iodine penetrate. Problem number two is that this leaf is dark green in color because it has lots of chlorophyll, and this dark green color will not make me see the color change very well. So I have to solve these two problems. Number one, I have to remove the waxy layer so that the iodine can penetrate the leaf. And number two, I have to remove the dark green color as much as I can. How do I remove the waxy layer? Look at the picture. How do I remove the
By-
waxy layer?
By boiling the leaf.
Very good, in water. I will boil my leaf in water. I will use a hot water bath because this is a safety precaution. I will not put my leaf directly on the flame, it will burn. I'm going to boil it in water. Why do I boil it in water? And this is a very important question that you will be asked in paper 6, because I want to remove the waxy layer. I want to destroy the cell wall and the cell membrane. I want to kill the enzymes inside the leaf. I want to kill the leaf. Why? Because when I remove the waxy layer and I destroy the cell membrane, the iodine can be able to enter inside the leaf. The iodine will be able to penetrate and enter inside the leaf. Do you understand why do I boil in water first? Anyone has any questions? Okay, no one is asking. After I boil it in water, I have still a second problem. My second problem is that my leaf is dark green. It's a very dark color because it has lots of chloroplast. So I want to remove the chloroplast as much as I can. I want to make this plant leaf light in color so that I can see the color change. So what do I do? Step 2, what do you see in the picture? This leaf will boil in what's
Ethanol.
this? Ethanol. So I'm going to boil my leaf in water to destroy the cell membrane and to remove the waxy layer so that my iodine can penetrate. Step number 2, I'm going to boil my leaf in alcohol or ethanol. Alcohol or ethanol are the same thing. And I'm not going to put the ethanol directly over a Bunsen burner. Actually, the Bunsen burner will be turned off, as you can see, because the ethanol is what, who will tell me? I think you took this in chemistry. Ethanol is very good. It is flammable, and this is a safety precaution. I cannot put the ethanol directly over the Bunsen burner. Even my Bunsen burner will be turned off because ethanol can burst in your face and it is dangerous. So I'm going to put my leaf in the ethanol and I'm going to put my test tube in the water bath, just as you see here. And I'm going to leave my leaf inside the ethanol for a couple of minutes because the ethanol will dissolve the chlorophyll and the chlorophyll will come out. Now, my plant leaf will not become white, but it will become very light in color. I removed most of the chlorophyll because it was too dark. And this is a problem. I will not be able to see the color change if it is too dark. So again, step number 1, I will boil my leaf in water. Why? To remove the waxy layer and to destroy the cell membrane so that my iodine can penetrate. Number 2, I'm going to boil it again, but this time in what? In what? In ethanol. I am boiling it in ethanol this time because I want to remove what?
The chlorophyll.
Very good. I want to remove as much chlorophyll as I can to make it lighter in color. And then step number 3, I'm going to soak it again in water, just dip it in water. Now, why do I do step number 3? Because when I put my leaf in ethanol, my leaf will be very hard. It will be very brittle. It will be like charcoal paper. It will be like dust. So I want to put it in warm water again, step number 3, to make it soft so that it bends with me. That's it. And then I'm going to put it on a white tile, on a white background. After I finish all that, I'm going to add my iodine and I'm going to look at the color change and see if it will change from yellow, brown to blue, black or not. Okay, so what are we doing? We are trying to test for the presence of starch because we will do a lot of experiments. At the end of the experiment, I want to see if this plant photosynthesized or not. How do I find out? If the plant did photosynthesis, this means that glucose was produced and glucose quickly is converted to starch to be stored. So if I want to do an experiment to see if the plant has starch or not, I need to do a few steps before I directly add the iodine on my leaf. I hope I'm clear. I'm going to slow down again and I'm going to repeat the question again. I have a leaf. Let me use green color. Okay, I have a leaf. This leaf is dark green. Now, I gave you this leaf in the lab and I asked you, please show me if this leaf contains starch or not. Can you simply add iodine directly on the leaf?
No.
No, I cannot add iodine directly on the leaf. I want you to pay attention because all this are paper 6 questions. Now, what are my 2 problems? I have 2 problems. What is problem number 1?
The
Waxy layer.
and cell membrane.
Very good. I have a waxy layer. What is the problem with this waxy layer?
It does not allow iodine
It does not let iodine penetrate through the leaf.
Excellent. The iodine will not penetrate. Now, how do I solve this problem?
boil the leaf.
Boil the
Excellent. I will boil it in water, in a hot water bath. So when I boil in water, what will this do?
dissolve the waxy cuticle.
Very good, but this is not enough. You will dissolve the waxy cuticle, you will destroy the cell membrane, you will destroy the enzymes, you will destroy the cytoplasm, you will kill the leaf. Why? Because when it is dead, only then will the iodine be able to penetrate. Very good. I hope problem number 1 is understood. Now, what is problem number 2?
Chlorophyll.
Chlorophyll.
Very good. It's dark. It contains lots of chlorophyll. I cannot see the color change. So how do I solve this problem?
Add the leaf to ethanol.
Very good. I will boil it in ethanol. Please, every word makes a difference. I will not just add it to ethanol. I'm going to boil it in ethanol. And boiling it in ethanol is not a safe step. I need to do some precautions. How do I boil my leaf in ethanol? Do I put it in ethanol This is my leaf. And then I put the test tube directly over a flame.
No.
No, we can do
No,
that
then-
because ethanol is flammable.
Excellent. Then what do I do?
Water bath.
Use a
Very
water
good.
bath.
Yes, I will put it in a water bath and my Bunsen burner will be switched on or off?
Off.
Off. Very good. So this is step number 2. And then I'm going to do a last step before I put my iodine. What is it?
Put
The
the
ethanol
leaf in
will
water.
make my
Leaf
leaf.
in water.
Very good. I'll just dip it in warm water to make it
Soft.
soft. And then I'll put it on a white tile and then my iodine. This can be a nice essay question that is very easy only when you understand it. Let's look at the pictures. You have all those pictures in your notes. These pictures make life very easy. Okay, step number 1, I'm going to boil it in water. And I think you know why. Step number 2, I'm going to boil it in ethanol. But I have to clearly mention that my Bunsen burner is switched off because it is flammable. And the reason, to remove the chlorophyll from the leaf, okay? Because the ethanol can dissolve the chlorophyll and we don't want it dark green, we want it a little bit lighter in color so we can see the color change. And then I will dip it in water again. This is my third and the step before the last to make it soft. And then the last step, I'm going to put it on a white tile and then I'm going to add my iodine. Why did I use a white tile?
Identify color.
In order to be able to see the color. Yes, it's always very good to see a color change when there is a white background. And then the last step, you will observe the color change. The color, if starch is present, will change from yellow brown to blue black. Did you understand? Did you understand what we said right now? If you understood, this is all what I want from you. I want you to understand the steps. I don't want you to memorize. Things like these should not be memorized. They should be very well understood. Okay, now there is a step known as destarching the plant. Again, I'm going to explain what destarching is and how it is done. So what are we talking about? We're talking about a procedure known as destarching. From the name, destarching means I'm going to remove all the starch from the plant. Now let's understand it together from the very beginning. The starch came from where? How did the plant make starch? Excuse me?
By
The
photosynthesis.
glucose.
Very
Uh,
good. Yes, you are correct. There was photosynthesis. Let's write down the equation together so that we understand it together. Carbon dioxide plus water. Now you are year 10. You have to know the balanced equation. In the presence of light and in the presence of chlorophyll will give me glucose. What's the formula for glucose?
C6H12O6.
C6H12, correct, O6 plus oxygen. Is this balanced?
No.
No. So how do I balance it?
Add 6 before carbon dioxide and before
Yes.
water, before oxygen.
Excellent. And before the oxygen, I will leave the glucose alone. So the glucose is what produced in photosynthesis, but we are testing for starch. Why are we not testing for glucose?
Because glucose is soluble.
Excellent. And it is highly reactive or not highly reactive?
It's highly reactive.
Yes. The moment the plant produces glucose, it is immediately converted to what and stored?
Starch, sucrose, cellulose.
Someone said sucrose, correct. The glucose will be transported as sucrose. By the way, this part is supplement, so it needs to be revised. Again, I think you all agree that glucose is produced or the product of photosynthesis, glucose and oxygen. Oxygen is released to the atmosphere. It is considered a waste product. It's a good waste product, the glucose. Glucose, it is going to be transported to different parts of the plant. How will it be transported? In the form of
Sucrose.
sucrose. Or it will be converted to fats, or it will be converted to proteins. Now, what if it is stored? It is stored in plants in the form of
Starch.
starch. That is why when we come to test if the plant did photosynthesis or not. We do not test for the presence of glucose because glucose is immediately converted. It doesn't stay as glucose inside the plant. It is converted to sucrose, it is converted to proteins, it is converted to fats, and it is converted to starch if it is going to be stored. That's why we test for the presence of starch, not for the presence of glucose, because glucose is soluble and highly reactive, and it is immediately converted into other forms. Do you understand this question? So if you receive a question in paper 4, why do we test for the presence of starch in photosynthesis experiment, not for the presence of glucose? I hope you can answer this question now. Clear? Any questions? Okay, going back to the de-starching part. De-starching, focus on the word de-starch. I'm going to remove the starch from the plant. If the starch was made by photosynthesis, then how will I remove the starch from the plant? I want you to
Respiration?
think. Okay, you are close. This means I'm going to make the plant respire only. This is what you
Don't
mean?
allow photosynthesis to happen.
What did you say? Can you repeat, please?
Don't allow photosynthesis to happen.
Excellent. You are 100% correct. De-starching the plant means I'm going to remove all the starch from the plant. Number one, we want to know why we will do that. Number two, we want to know how this will be done. Let's go to how first.
You keep
because
it in
how
a dark area.
Excellent. De-starching means I will remove all the starch from the plant. How this is done? I'm going to prevent photosynthesis. Just like you said, I'm going to put it in a dark area for 2 to 3 days only. And why am I stressing on only? What if I keep it in a dark area for 2 to 3 weeks? What will happen?
Will die.
It will die. Exactly. I'm going to put it in a dark area in a cupboard, or I will cover it with aluminum foil. I'm going to prevent photosynthesis for 2 to 3 days only, so that the plant will respire only. It will not photosynthesize, and it will use up all these stores of starch. It will finish up all the stores of starch. So how? I'm going to put it in a dark cupboard to prevent photosynthesis. Now the question, why do I do that before photosynthesis experiments? What do you think? Why do I de-starch the plant?
To
Think
know
with
how
me.
much starch is produced during photosynthesis?
Yes, you are correct. Now let's say I have a plant that I took from the garden and I did an experiment, any experiment, and then I test for the presence of starch. If I do not de-starch the plant, every time I will have positive results. Why? Because the plant that I took from the garden already has starch. I should remove the starch before I start to see if starch will be produced during my experiment or not. If I do not de-starch the plant, then every time I do the experiment, I'm going to receive a positive result. Who does not understand this part? Tell me. I will try to explain it again. For example, 1 minute, let me, excuse me. Okay, we are talking about de-starching. Again, de-starching we are talking about de-starching. One by one. De-starching means I want to remove all the starch from my plant. Why do I do that? Two, how do I do that? Now let's talk about why do I do that? Why do I de-starch? Because I want to do an experiment. Let's say I want to put my plant in an environment where there is no carbon dioxide, and I want to see if it will photosynthesize or not. After I finish my experiment, I'm going to see if it photosynthesized or not. How do I know it photosynthesized? If it did photosynthesis, what should it produce?
By the presence of starch.
Starch.
Exactly. If this plant photosynthesized, it should produce starch. So at the end of my experiment, I will test for the presence of starch. If starch is present, then this plant was able to photosynthesize without carbon dioxide. If starch is not present, then this plant was unable to photosynthesize in the absence of carbon dioxide. But I already took this plant from my garden. This plant already had starch. So before I start my experiment, I have to do what? I have to de-starch my plant. I have to remove all the starch and then do the experiment and then do the test to see if starch was produced during my experiment or not. Because if I do not de-starch my plant, every time I'm going to receive positive results. So I de-starch my plants before plant experiments so that I know if starch was produced during my experiment or it was not produced during my experiment. In other words, to receive accurate results, but I have to explain what accurate means here. Who does not understand? Or who wants me to repeat this part? So now, is de-starching clear? Okay, you're not telling me that it's not clear, so I assume it is. So let's continue from where we stopped. De-starching the plant, we will have to de-starch the plant by putting the plant in a dark room for 2 days so that all the starch is removed, so that the plant is not allowed to do photosynthesis. Why do we do that? So that we know that the new starch that has been produced will be produced during the experiment, and it was not there from the beginning of the experiment. So I want you to understand this sentence. De-starching the plant ensures that any starch already present in the leaves will be used up and will not affect the results of the experiment. So your results of the experiment are accurate. Okay? You should test for the starch produced during the experiment, not for the starch that was present before the experiment. Anyone has any questions?
Can you repeat the part of the it will not affect the result of the experiment? How?
Okay, so this shows me that you don't understand. Again, my dear, I have a plant. Okay? This plant was in my garden. So it was exposed to the sun and it was photosynthesizing. This plant, does it have starch or it doesn't have starch?
It does.
It does. Okay, so starch is present. I'm going to take my plant and I will expose it to a particular experiment, any experiment. I'm going to remove the magnesium, I will remove the nitrates. I want to see if this plant will photosynthesize during my experiment or not. So I am doing my experiment regardless of what my experiment is. But before I do my experiment, I did not remove the starch. The starch was already there. I did not de-starch. After I finish my experiment, I'm going to test for the presence of starch. I will see if starch is present or not. Of course, my results will always be positive because did I remove the starch before the experiment?
No.
No, it was already there. So what happened during my experiment? I don't know. The starch was already there before my experiment started. I have to de-starch before my experiment to see if starch was produced during my experiment or not. Do you understand now how my results will be accurate only if I de-starch the plant first?
Yes, I understood. Thank you.
Okay. I hope it's clear. Okay, so all the experiments that will follow from now on, we have to de-starch first. And the experiments that will come from now on want to confirm a certain hypothesis. We have a certain hypothesis. We are convinced that carbon dioxide and water and light and chlorophyll are essential for photosynthesis. C6H12O6 plus oxygen, and this is nice and balanced. So this is the photosynthesis experiment. We are convinced that we need carbon dioxide, and we need light, and we need chlorophyll for the making of glucose and oxygen, for the success of photosynthesis. So we will carry on certain experiments. In every experiment, we will remove one of those factors. Number one, we will remove CO2, and we will see if the photosynthesis will happen or not. Number two, we will remove the light in another experiment, or we will change the intensity of the light. In another experiment, we will remove chlorophyll. And we will see if we remove one of those, photosynthesis will happen or not. We usually never use an experiment in which we remove the water, because water, if it's not there, the plant will die. But before we start the experiment, we have to do what?
Destarching.
Destarching. So going back to our investigation questions, we start our question by my independent variable is, my dependent variable is, my steps are. We will write the same thing, but from now on, any experiment that involves plants, before I start all my steps, I will write one sentence. I will destarch my plant. And you don't even need to explain why you do that, except if they ask you. I will just destarch my plant so that my results are accurate. That's why I'm telling you, I don't want you to memorize. I want you to understand what's happening. Let's go back to the experiments. The experiments are very easy. Experiment number one. Is light necessary for photosynthesis? We want to know the answer to this question. We got a leaf, okay? And we destarched it, of course, before we put it in a cupboard for 2 to 3 days. So we removed most of the starch, and we did not allow it to photosynthesize for 2 or 3 days only, not more than that, because we don't want to kill the plant. We only want to destarch the plant. And then we covered part of the leaf with aluminum foil. Now, why did we cover it with aluminum foil? Who will tell me?
Because it doesn't allow the sunlight through.
Very good. It will reflect the sunlight. Please use the word reflect. Okay, because aluminum is shiny and it will reflect the sunlight. And we will leave it in the sun for a couple of hours. And then after we finish, we will test for the presence of starch by using iodine. And don't forget, before we test for the presence of starch, we will boil it in water to remove the waxy layer and destroy the cell membrane and make it permeable. And then we will boil it in ethanol to remove most of the chlorophyll. And then we will put it back in warm water to make it soft. And then we will add the iodine. Now, when we add the iodine, which parts will give me a positive results? The parts that were exposed to the sun or the parts that were covered with aluminum foil?
The ones that were exposed to this.
Yes, the ones that were exposed to the sun, of course, will give me a positive blue-black color because they were exposed to the sun. And the ones that were covered with aluminum foil will give me a negative result, and the iodine will remain yellow-brown. Okay, now I want to ask you a question. If we did this experiment before, but we did not destarch, like we started immediately, what will be the results?
All the leaf will be blue.
All of them will be blue-black.
Exactly, excellent. The whole leaf will be blue-black, even though we covered part of it, but that part already had starch from before and we did not destarch, okay? Now, if they're asking you for the independent variable here, what's the independent variable? What are we testing for, the presence or the absence of?
Right.
Light. And the dependent, the results, which will be the presence or the absence of starch. We will see if starch is present or not. Okay, please make sure you understand. I don't want you to memorize anything. Another experiment. In this experiment, we will remove carbon dioxide. We will ask ourselves, is carbon dioxide needed or not needed? We will see. Step number one, I'm going to destarch. I'm going to put it in a cupboard for 2 to 3 days so that all the starch is gone. Step number two, I'm going to do my experiment. Step number three, I will test for the presence of starch. Let's see how I will do my experiment. I want to remove the CO2. I'm going to put my nice plant and cover it with a plastic bag, as you can see. And then I'm going to use a material known as soda lime. And again, I do not want you to memorize it. They will tell you in the question that they will put inside this bag soda lime. Soda lime is a chemical that absorbs carbon dioxide from the atmosphere. So all the carbon dioxide will be sucked. So this plant will be in an environment where there is no carbon dioxide. I will put it in the sun and I will give it time to photosynthesize. I will see if it will photosynthesize or not. After I leave it in the sun for a couple of hours, I'm going now to test for the presence of starch. I'm going to take one of the leaves, boil it in water to remove the waxy layer and destroy the cell membrane, and then boil it in ethanol to remove most of the chlorophyll so that I can see the color change, and then dip it again in warm water to make it soft. And then the last step I will do, I'm going to add my iodine and look for the color change. If the color changes from yellow-brown to blue-black, then this means the plant photosynthesized or did not photosynthesize. If there's a positive result,
Photosynthesize.
photosynthesize. But I want you to tell me, what do you expect when there is no carbon dioxide? Will the plant be able to do photosynthesis?
No.
No. Okay, the equation says I need carbon dioxide and I need water. Without carbon dioxide, I cannot do the glucose. I cannot do photosynthesis. So when there is no carbon dioxide, there will be no photosynthesis. I should get a negative result. Okay, now this is another experiment. This experiment, we want to know, is oxygen gas produced during photosynthesis or not? So what did we do? We got a plant. This plant lives in water. It is adapted to photosynthesize underwater. And then we set up this experiment. We put an inverted funnel on top of the plant. And then we put here small blocks of wood to lift the funnel so that the water is able to circulate. And there is a test tube here, as you can see, and it is all inside water and it is exposed to sunlight. This plant should do photosynthesis and it should release oxygen as a waste product. When oxygen is released, I will see what?
Bubbles.
Bubbles. So bubbles is an indication that a gas has been produced. Now here we have lots of errors and so many ways to modify and lots of nice questions. Now counting bubbles, is it accurate?
No.
No, it is not accurate. Now why?
Because you may
Because
count.
bubbles are in different sizes وكده.
Very good. Bubbles can be of different size. Bubbles can bubble very quickly. I can miscount. The bubbles are different sizes. Put in mind, please, that everything you write is a mark. So the bubbles can be of different sizes, so it's not accurate. Maybe I will not count the number of bubbles correctly. The bubbles are being produced too quickly, so I cannot count. So how can I modify?
Gas syringe.
Very good. I will measure the volume of the gas produced, not count the number of bubbles. So instead of counting the number of bubbles, I'm going to connect this apparatus to something with a ruler, a graduated gas syringe to measure the volume of gas, connected to an inverted cylinder, anything with a ruler to measure the volume of gas, but not count the number of bubbles. You will see this question so common in paper 6. Okay, another question. How can I confirm that the gas that is being produced is oxygen and not any other gas? What is the test for oxygen? Who knows?
Glowing splint.
Yes, I hope you took it in chemistry. It will relight a glowing splint. What is that? The best thing to do is to show you a video. 1 minute. Let's go to YouTube. Okay, because where is YouTube? 1 minute. No, this is not what we want. Okay, test for oxygen relight No, this is not what we want at all. Okay. Do you see how it relit? This is relighting a glowing splint. If oxygen is present, the fire will relight. Relighting a glowing splint. Did you all see that? Okay, did you see it with your own eyes? Did you see how it relights a glowing splint? Okay, so this is one experiment. We want to know if oxygen is being produced or not. We will put my plant, and first we will destarch. No, it's not, we will not destarch here. Why won't we destarch here? Are we testing for starch at the end of the experiment? No, so we're not testing for starch, so we will not destarch. We will take the plant, we will put it in water, and we will collect the bubbles. And then we want to make sure that these bubbles are oxygen. So there is a test for oxygen. We will get out the test tube quickly, and we will insert in this test tube. What do I insert?
Glowing.
Very good, a splint. A splint is a wooden bar, like you saw in the video, that is already lit. But if there is oxygen, it will relight or re-glow. This is the key word. In the presence of oxygen, oxygen makes fire relight or re-glow. So this test should be a positive test because oxygen is produced in photosynthesis. Okay? Now, all the 4 experiments show us that starch formation, which is our evidence for photosynthesis, does not take place in the absence of light. When we remove the light, we have a negative result. When we remove chlorophyll, we have a negative result. When we remove carbon dioxide, we have a negative result. And also our glowing splint, when it relit, it showed us that oxygen is produced. There is an experiment that I think we missed. This experiment. This experiment here is going to test if chlorophyll is needed for photosynthesis. Number 1, I picked a leaf that looks like this. It's called chloriated or variated leaf that is part white and part what? Green. The green part of the leaf shows you that it contains chlorophyll or it does not contain chlorophyll.
Does not contain chlorophyll.
The green part, it has chlorophyll or it does not have chlorophyll?
It has chlorophyll. The
It
green
has
part has
chlorophyll.
chlorophyll.
Very good. So I'm going to choose a leaf. Number 1, I'm going to destarch it. Don't forget this, please. It's just a sentence, but it makes a big difference. I have to destarch it. I will put this leaf in a dark cupboard for 2 to 3 days to remove the starch. Step number 2, I'm going to carry out my experiment. I'm going to put it in the sun. I'll put it back again in the sun because I want it to photosynthesize. Step number 3, I will test for the presence of starch. And step number 3 in itself is made up of several steps because I have to boil it in water first and then boil it in ethanol and then dip it back in water. I think you understand the reasons why. And then I will look at my results. The part that was originally green will give me a positive result or a negative result?
Positive result.
Positive result. So this shows me that chlorophyll is needed for photosynthesis. And the part that was white will give me a positive or a negative result?
Negative.
The negative, yes. Put up your voice. You are all giving me correct answers, but be sure of yourself. So this shows me that when it was green, it gave me a positive result and photosynthesis took place. And when it was white, it gave me a negative result and no photosynthesis took place. So these results show me that chlorophyll is needed or not needed?
Needed.
Is needed.
Needed. Okay. I hope you understand the experiments. This is a very simple experiment. Again, I will destarch and then I will put in the sun. Before I put in the sun, I will cover part of it. That part that was covered will give me a negative result. And this shows me that light is needed for photosynthesis. Very easy and simple experiments. This experiment, I'm going to see if carbon dioxide is needed or not. I will get my plant. First, I will destarch. And then I'm going to put it in a plastic bag that contains soda lime. Soda lime will absorb or suck the carbon dioxide. I'll put it in the sun and give it a chance to photosynthesize. And then I will see if photosynthesis happened or it did not happen. How do I find out? If it made starch, then photosynthesis took place. If it did not make starch, then photosynthesis did not take place. And I do expect that if there is no carbon dioxide, it will give me a positive or a negative result?
Negative.
Negative. Okay? And don't memorize those molecules or those compounds. In case you want to know, soda lime absorbs carbon dioxide and sodium hydrogen carbonate provides carbon dioxide. But they always tell you in the question. In this experiment, I want to know if the gas produced is oxygen or it is not oxygen. So I'm going to do the oxygen test. What is the oxygen test?
A splint.
The
Relight
splint, I'm
advancement.
going to put a splint, and if it relights or reglows, then it's oxygen. Anything hard in what we said today? Let's explain this last experiment and then I'll let you go. This experiment wants to study the effect of a changing light intensity on the rate of photosynthesis. So what is my independent variable?
Light intensity.
Intensity of light.
Very good. I will change the light intensity. Now, how do I change the light intensity? Look at the picture here and try to figure out how did we change the light intensity?
Changing the distance of the glass tank.
Yes, you are correct. Light intensity means that one time I'm going to use strong light and another time I'm going to use weak light, and I'm going to see how this affects the rate of photosynthesis. Now, I want to talk about several points. What did I do? I got a water plant. This is my aquatic or water plant. It's a plant that can live in the water. And I will put it in my water beaker with an inverted funnel, as you can see, and this funnel is raised a little bit so that water can circulate. And I'm going to put it in the sun, and I'm going to give it time to photosynthesize. Okay, and then what do? I will not de-starch here because I'm not going to test for starch. Wait and see. And then I'm going to put a lamp here. This lamp is at a certain distance. Let's say it is at 80 centimeters. And I'm going to count the number of bubbles produced over a certain period of time. Let's say 50 bubbles were produced in 2 minutes. And I'm going to repeat the experiment. But this time, I'm going to put my lamp a little bit far away. Here, I'm going to put it here. So what am I changing? In order to change the light intensity, what am I changing? What am I playing around with?
Distance.
Excellent. So every time I am increasing the distance, and every time I am changing the distance, I am counting the number of bubbles produced over 2 minutes. So the 2 minutes are constant. Okay. Why is there a water bath here? There is a beaker here that is filled with water. Why?
To filter off heat given off by the lamp.
Yes, those that took this experiment last year will know the answer. Because this lamp is releasing light, but also with light always comes heat. And this heat can affect my results. So we want to put here a water beaker to absorb the heat. This water beaker will act as a heat filter. Okay, now, if I want to design a table, because you will be asked to design a table, what is my independent variable?
Light intensity.
Sense of the light.
Okay, the intensity of the light. How was this changed?
The
What did we change?
distance.
Exactly.
The distance.
Excellent. So on the left side of the graph, I put the independent variable or the dependent variable?
Independent
Excellent. Which will be what? What did I change? You just said it.
distance.
Excellent. I'm going to change my distance. So the distance will be put here on the left side of the graph. And don't forget the units, the distance in centimeters, for example. Now, and then what will be my results?
Volume of gas produced.
You are correct. Either the volume of gas produced or the number of bubbles produced over in 2 minutes. But what is more accurate, volume or the number of bubbles?
Volume.
Volume, very good. So I'm going to write here volume produced in centimeter cube, don't forget the units, in 2 minutes. And I'm going to change the distance, let's say 5, and the volume, let's say 10, distance, whatever, and the volume 15. No, the volume, what do you think? The volume will increase or decrease if I increase the distance?
The volume will decrease.
Correct. The volume will decrease because less photosynthesis. Now, I have several questions for you. How can we calculate the rate of photosynthesis?
Over time, distance over time.
Excellent. Rate is always a particular measurement over time. I don't know if you took physics last year. For example, speed. Speed is distance over time. Rate is always a particular measurement over time. So how will we calculate the rate? It will be the volume of the gas produced over, what's the time?
2 minutes.
2 minutes, correct? Okay, these are the questions, by the way. The questions don't come out of that. But a lot of you don't know what's the rate. A lot of you do very small mistakes that we want to avoid. So I hope this experiment is clear. Let's get the experiment again. Here is a more accurate experiment, but this experiment has one mistake. Again, this experiment is changing the light intensity and seeing how the light intensity change will affect the volume of gas produced. So I have a ruler here to measure the distance, and every time I am changing the distance. And here I have the pondweed in my water, and the gas we here will be measured using a what's this?
Gas cylinder.
Very good. Is this accurate or not accurate?
Accurate.
Accurate. But there's a mistake here.
A glass
It's
tank.
missing a water filter.
Yes, this glass lamp releases heat, and this heat can affect my results. So I have to put here a water filter, just a beaker that has water to absorb the heat. And by the way, I can use this same apparatus if I want to change another independent variable. For example, the what? Temperature. I'm going to fix the distance of the light. So the distance will not change. Nothing will change, but I'm going to change the temperature of the water if I want to study how the temperature will affect the rate of photosynthesis. How do I change the temperature of the water?
Thermostatically
Decrease the
controlled water bath.
Very good. Thermostatically controlled water bath. Okay? So the experiments, believe me, once you understand them, they become very easy. Here, investigating the effect of changing temperature by changing the temperature of the water in the beaker. So here we either use, this is a pan balance or a thermostatically controlled water bath, just like you know it. The independent variable here will be the change of temperature, and the dependent variable will be the rate or the number of bubbles produced over 2 minutes. Here is the glass tank. This experiment is so common, and it is very nice that it is common because it's very easy. And I can change any factor. I can keep the distance of the lamp fixed, I can keep the temperature fixed, and I can change the concentration of carbon dioxide. Maybe I can use 1% carbon dioxide by using the sodium hydrogen carbonate. You don't have to know the name of that compound, but I can change the temperature by changing the water bath. I can change the light intensity by changing the distance of the lamp. I can change the carbon dioxide concentration by changing the amount of carbon dioxide present in the water. I can play around with the independent variable, and in the end, the results are the same, or I will use the same method to obtain my results. I will count the number of bubbles or measure the volume over a fixed period of time, and from there, I will be able to calculate the rate of photosynthesis. Okay, I promised you that I will stop, and I will, promise. Now, if The light intensity or the distance of the lamp is my independent variable. Where do I plot it? On the x or the y axis?
X-axis.
Yes.
Axis.
So the distance will be on my x-axis, and this distance, don't forget the units like you always do, in centimeters. And what will be plotted on the y?
Volume of oxygen.
Yes, the volume of oxygen produced in 2 minutes, or if they want you to calculate the rate, then the rate will be on the y-axis. It depends on the question. And we will plot our graph nicely, equally distributed. You are now really good at doing graphs. But some of you, I don't know, I saw this in the term exam. Some of you, they make a graph inside a graph. Like, for example, you already have the grid, and then they draw a line like this, and then a line like this, and then they create a graph inside the graph, and of course they get all the axes incorrect and they lose lots of marks. I don't know why do you do that. Please don't draw a graph inside the graph. Okay, I promised you I will stop, so I will. The experiments that we discussed up till now, are they clear? Okay, I hope they are. Please read what we took. You are now during your holidays and you have time. Read. I want you to understand what we took. We stopped at the limiting factor. This is supplement, so this is important. And this is what we will start with next time. I'll meet you on Saturday like we always do at 1 p.m. We will go back to Saturdays. Okay? And you have homework for those who are asking me for homework. Finish drugs, paper 2 and paper 4. For those who are amazing and they already finished it and they want to solve more, if you want to, you can start paper 6 plants because it's too long. If you want to. Okay, any questions before I go? Is everything said today clear? Okay, no one is asking questions, so have a good day and start solving your homework. Bye.